Hello, good afternoon, everyone. Okay, it's nice to be back. Thank you for watching my channel today. Thank you for joining my class. Okay, I do not know who is with me here. Actually, I like to say apologies. Okay, for last week I was not able to have a class because I was not feeling well. So today I'm back, and I hope that you'll be able to join in the class and learn what you're supposed to learn. All right. So today I would like to continue with chapter 3, which is 3.4. Sorry, 3.3. Okay, next week I will do 3.4. And then hopefully we will be able to start chapter 4 soon. All right, because form 4 is actually very long. The syllabus is very long. So we got to keep on moving. Yeah? Otherwise, we will not be able to finish the syllabus by the end of the year. Okay, so I'm going to share screen now. I do not know who is with me, but if you'd like to let me know, please do drop me a line at the comment box here. Okay, right, let me just share screen. I hope you can see this. All right, so today I'm going to do movement of substances across the plasma membrane in living organisms. Okay, so this is continuation of chapter 3, where we learn about the different types of methods of transport. We have active transport, and then we have the passive transport, which comprises osmosis, simple diffusion, and also facilitated diffusion. So today we're going to look at some examples of these types of transport, okay, in living organisms. All right, okay, let me just look at my own screen. All right, so, okay, so this is what, okay, this is the first screen here, the first, okay. So these are the examples, and what are the examples of active and passive transport in living organisms? So you need to remember every method of transport, you need to remember at least 2 examples. Okay, so here I've given, this is straight from the textbook, yeah. Okay, so from the active transport, you need to remember a few. So here there are 4 examples. They all include the osmosis, simple diffusion, and also facilitated diffusion. Whereas for the active transport on the right hand side here, they are all examples of active transport. Okay, let's look at the first one. Yeah, after I'm going to show you each one of them using diagrams. Okay, first one is gaseous exchange between alveolus and blood capillary through simple diffusion. This one you have learned in Form 3 before. When you learn the inhaled air enters the respiratory tract until it reaches the alveolus. Okay, after that you find the oxygen will diffuse across the alveolar membrane into the blood vessels and blood capillaries, and at the same time carbon dioxide diffuses out from the red blood cells into the alveolus. So this is an example of actually simple diffusion. Okay, so the gases exchange between oxygen and carbon dioxide is through simple diffusion. Okay, next one, reabsorption of water. Okay, so now we must remember the word reabsorption, not just absorption. Yeah, okay, we do not accept the word absorption of water. Okay, when we want to produce urine, right? A lot of the liquid that passes through the glomerulus, through the kidney tubules, we call the renal tubule. Okay, most of the water is actually reabsorbed back into the blood. Okay, because we do not want to excrete too much water from our body, because water is important. Okay, we have to take back some of the water. So that process of reabsorbing back water into the blood, okay, from the kidney, that process actually uses the process that is under osmosis. So every time remember the word reabsorption of water or uh, diffusion of water, it is actually osmosis. Okay, so that's just like osmosis is a very fancy word for the absorption of diffusion of water. Okay, next, absorption of water, okay, by plant root hair cell by osmosis. This one is also true, osmosis. As long as it has to do with absorption of water, or whether reabsorption or diffusion, as long as water movement, if you remember, okay, your previous lesson 2 weeks ago. So any movement of water molecules is actually through osmosis. Okay, it's through osmosis, nothing else. Don't talk about simple diffusion. don't talk about facilitated diffusion or or active transport. It is always osmosis when you talk about diffusion of water. Okay, one more example under passive transport is absorption of fructose molecules in the villus by facilitated diffusion. Okay, because this one requires carrier protein. Okay, because fructose is a large molecule and fructose is water soluble. It is not lipid soluble. If it's lipid soluble, it can go through the phospholipid bilayer easily. Okay, but because this is not fat soluble, it's water soluble, it cannot enter the cell through the phospholipid bilayer. Therefore, it has to go through the uh, carrier protein. Okay, and that requires this transport protein. So that is called facilitated diffusion. Okay, so next one, let's look at examples of active transport. Okay, now this one is absorption of glucose and amino acid in the villus, because this one happens against the concentration gradient. Whenever you talk about active transport, you have to remember that it has to be from a region of lower concentration of this substance moving into another area which has a higher concentration. That means inside, or whether this region here already has a lot of this molecule, but the cell still requires it. So it is able to move in even though it has got less outside, okay, from A to B. From A is less, B is more, but it's still able to bring it inside because it's using energy to do it. Okay, so next one, the absorption of glucose. Okay, now this is not water, yeah. If you notice, you compare with the other side. Just now we mentioned on the left hand side, reabsorption of water by osmosis. But this one, reabsorption of glucose, okay? Glucose is also against concentration gradient, okay? Inside the renal tubule, all right, you will have uh, the water, the liquid that goes into the renal tubule will have some glucose, but we do not want to excrete the glucose. We want to absorb back the glucose into the blood. So even though the blood has got more glucose, all right, the body still requires the glucose, so it can move in back into the blood from less to more through active transport. Okay, next one. Transport of sucrose from a leaf to a phloem tissue. This is what we call transport of organic substances through the plant. Okay, when you learn in form 2, form 3 before, xylem is the tube that the vascular tissue that transports water in the plant. For uh, for food, it goes under it's transported through the phloem. So the process of transporting food, okay, sometimes it is done through active transport, okay, because from a region of less, okay, concentration, it is pumped into a part that has got more, okay, where the food is accumulated. So it also goes against concentration gradient. Okay, one more. Absorption of mineral ions. This one is always active transport. The method by which plant absorbs mineral ions from the soil into the cell, the root hair cell, it is through active transport. Okay, not water. Water is always osmosis. So this mineral ions absorption is through active transport. All right, okay. Now let's look at it again with examples. So this is example of simple diffusion. This one I mentioned about the alveolus. Okay, oxygen diffuses from alveolus into the blood capillary through simple diffusion because there is higher concentration of oxygen inside the alveolus compared to the oxygen inside the red blood cells. Okay, because this comes from the body cells. Right, this blood that comes to the alveolus here is without oxygen or deoxygenated blood. So therefore, there will always be more oxygen in the alveolus compared to in the blood. Therefore, oxygen will diffuse inside. Okay, and then at the same time, right, carbon dioxide will diffuse from the blood capillary, so it has a higher concentration of carbon dioxide, and it goes out through the alveolus through simple diffusion, and that's how we exhale, right, this carbon dioxide. Okay, and this is due to the concentration gradient. Okay, the movement, whether it goes in from A to B or B to A, all depends on which side has got more, which side has got less. So this one follows the concentration gradient, always from more to less. Okay, whether it's oxygen or whether it's carbon dioxide, this is through simple diffusion. Okay. All right. Okay. I think there's no question here. So simple. Now, question. Second example here is an example of osmosis. So you will see this important word here, water. As long as we're referring to absorption of water, it's always osmosis. Okay, doesn't matter moving from where to where, right? It's always from water is always osmosis. Okay, another example number 3 here is reabsorption of water. So you see all this? Whenever you see the word absorption, reabsorption, diffusion, it's always through osmosis. Okay, now I will not go into detail about the renal tubules because you only see it, learn it at the end of the year. Then you will see in detail lah. You just need to know that our body will just take in back water, okay? Because we do not want to excrete so much water. Otherwise, our body will be dehydrated. We need to excrete water, of course. That is just enough to dilute the mineral salts and so on. But the majority of the water is actually reabsorbed back into our blood. Okay, otherwise we'll be losing too much water. Okay, now example number 4, this is under passive. The example number 4 is on facilitated diffusion. So this is the small intestines, okay, in our body, and this is one of the cross section of the small intestine. You will see that they are highly folded. Okay, inside the small intestine, you have these finger-like projections. You will learn that in chapter 6 later. So these finger-like projections, like jari-jari, yeah, all these jutting out here, they are to increase the surface area. They are called villi, okay, or villus or villi. And this shows one of those finger-like projections, which is called a villus, and it is the function here is to absorb. Okay, it is to absorb the nutrients that have been broken down. Okay, that have been digested. So it goes into the blood or it goes into the lacteal. Okay, to be utilized by the body. So this number 4 here is example of absorption of fructose molecules into villus by facilitated diffusion. Because fructose molecules are large and they are water-based or water soluble, they have to go into the cell or into the blood capillary through the epithelial cell here, and it cannot go through the membrane. Okay, it has to go through the carrier protein. So when you use carrier protein, this one definitely is facilitated diffusion using this either carrier protein or pore protein or channel protein. Okay, right. Next. Now let's look at example of active transport. Right, just now was passive, now active transport. Examples of active transport. So remember, you need to remember 2 examples at least for every type of method, so that when you write your essay or when you're asked to give examples, anytime you're able to cite an example. Okay, that strengthens your answer. All right. Okay. So this is the same picture that I showed you just now. It looks like the same thing, but we're talking about different substance here now. Just now we're talking about fructose, yeah? Okay, the absorption of fructose through the facilitated diffusion. Now we're referring to the absorption of glucose and amino acid into the villus by active transport. Okay, that means outside the villus here, this one here, let me look at this. Okay, so here you have glucose. Right now. Here you have glucose. Oh, sorry, it is wait. Oh, sorry, I have to reopen my PowerPoint. Yeah, there's a glitch here. There's a small little glitch. It's processing. It's opening again. Sorry, yeah. I was trying to write something on it and then it didn't work. Just 1 minute. It's opening again. Let me just open it now. Okay, so wait, 1 minute. I am going through the same, going through the slide now. So I don't want to repeat the slides that we have been looking at. Okay, this one. Slideshow and one slide. Okay, let me look at this again. So sorry. This is like glitch here. I've got 2 open. Okay, I've got two slides that open. Okay, so sorry, I'll open again. Yeah. Share screen, window. Okay. All right. Okay. So this is chapter. Oh, I've already opened the wrong one. I'll go through. I'll go through this one. A minute again. I'm so sorry. This what happens when you got problems with your slide. Yeah. So sorry. Yeah. Okay. Sometimes we don't plan these things. Yeah. Okay. All right. Let me go to this slide. Okay. This slide. This slide. So okay. I think I'm ready now. Window. Okay. Yeah. So sorry. Yeah. Right. Okay. So now, let me go through this again. Just now I was trying to tell you. Actually, I was trying to draw this thing. Okay. This is glucose. Let's say this is glucose molecule. Okay. Glucose molecule wants to go inside there. All right. Okay. So here is actually less concentration, lower concentration compared to what is already in the blood. But it doesn't matter because your body needs to have more glucose. So your glucose, even though here is lower concentration, it is able to go through the cell and enter the blood capillary. So you have to do it by active transport. It goes against the concentration gradient. Same with amino acid. Okay, it will enter into the bloodstream against the concentration gradient. Okay, now example number 3, mineral ions. Okay, they are absorbed into the root hair cell through active transport. So here we are referring to mineral ion. It looks like the same picture, right? It is the same picture, but we're talking about different substance here. We're talking about different substance here. The different substance here is just now was water. When you talk about water, it is osmosis. But here is mineral ion. So mineral ion is referring to active transport. Okay, so this is active transport. Okay, and here this example number 2, okay, is the absorption of glucose by renal tubules in the kidney. So blood, renal means the glucose will also be reabsorbed back into the blood. Okay, so we're talking about here lah. Okay, you can't see it here, but the blue tube, the tubes that are red, this is blood. Okay, so glucose will be absorbed back into the blood. Okay, the renal tubule are the yellow color tubes there. Okay, so that one has got glucose. It has passed by, it has gone into the renal tubule, and now we're taking it back into the blood. Okay, example number 4 for active transport. Transport of sucrose from leaf cell to phloem tissue. Okay, so you see the one in green here is going to the leaf cell. So as you know, leaf cell is the cell that produces food. Okay, it carries out photosynthesis. So the food is actually made inside the leaf cell, and it's not stored there because it has to be transported to other places of the plant that does not make food. For example, flower, okay, or the roots, or any part of the stem, or any other areas. So it has to go through the phloem. Okay, this process of transporting food, okay, through the phloem is called translocation. Translocation, okay. So sucrose is transported from the source. The source is the leaf cell, is where the food is made, okay, to the sink. The sink is where the food is going to be kept or used. Okay, so this is the source, all right, and it goes into the phloem tissue, and then this is the sink. The sink is like, for example, root cell or flower cells or any other part of the plant that does not make food, and this is where the food is transported to. Okay, so this is through active transport against concentration gradient. Okay, now let's go on to one important part of the concept about the different types of solution that we're going to look at later. All right. Throughout until the end of Form 5, make sure you have to remember this, the different types of solution in terms of concentration. So we have special names, right? So there are three types of solution here depending on the difference in concentration. So the first one, we look at the word isotonic. Iso. Iso actually comes from the word which means same or equal. Isotonic, yeah. Iso is means same. Okay, like you have your isosceles triangle. Okay, the triangle which has got two sides which are equal in length, right? Okay. Then the second one is called hypotonic. Hypo means under or below. Under or below, right? That means lower. Okay, then the other word hypertonic. Hyper means above. Okay, I'm sure you've heard the word hyperactive people, right? Hyperactive kids. Kids are too active. Okay, they jump about. Okay, they be bouncing off the walls, right? That's hyper. So above or over. Okay, now so there are three names here you have to remember. And let's look at the definition. Yeah. Okay, what is an isotonic solution? Okay, so this word solution, the word isotonic refers to a type of solution. Okay, depending on you compare this solution with another solution. So you have to be comparing this solution to another solution, then only you know whether it is isotonic, hypotonic, or hypertonic. Okay, definition. The solution is said to be isotonic when it has the same concentration as another solution. So if it has the same concentration as another solution, we will call it as isotonic. Excuse me here. I need to drink some water, please. Okay. So the concept here is isotonic solution. Now, if both solution A and B, if they separated by a selectively permeable membrane in the middle, right? You find that both solution has the same concentration of solute. So how do you know? Yeah, you can usually if they draw circles to represent the solvent or the solute, you can count. You count the number of the circles here. For example, the red one here represents the solute. Okay, you can see here A, it has got two solids here on the left, and for B, two solids. Okay, the number of solids look like the same. Now, then you count the number of solvent. Solvent are these water molecules. Okay, the red one probably they are sucrose. Okay, sucrose molecules. So the one that is blue here, they are the water, they are the solvent. So let's count 1, 2, 3, 4, 5, 6, 7, 8. Yeah, 8 on the left. 1, 2, 3, 4, 5, 6, 7, 8. So you can see on both sides of the solution, the number of solute is the same and the number of solvent is also the same. So they both have the same and equal concentration. So you will say A is isotonic to B. Or you can also say solution B is isotonic to solution A. Both have the same concentration. Okay, so solutions A and B are isotonic towards each other. So because the number of solids are the same, the concentration is the same. So when you talk about water, only water can move across the selectively permeable membrane, okay, because the water molecule is small. The solid is too big. They are actually small little holes or pores on the selectively permeable membrane. Okay, so it cannot pass through. The small little pores there cannot allow the big solid to pass through. So because of the concentration is the same, so the amount of the number of solid that passes through from A to B will be the same as B passing to A. So there's no net movement. How much that goes from A to B will be the same as how many molecules that go from A to B or B to A. So there's no net movement. It's equal on both sides. Okay, so 10 passes through, and then on this side 10 again passes to the other side. So there's no change in the total number of molecules or the solids. Okay, so that's called isotonic. So A is isotonic to B. At the same time, you can also say B is isotonic to A. Right? Okay, now next one. Let's look at hypotonic. Hypo means under, okay, or less. Right. So A solution is said to be hypotonic to another solution. So you always compare with another solution, yeah? When it has a lower concentration to the other solution. When it's lower, okay, less concentration. That means you must refer to the solute. When you talk about concentration, usually we are referring to the solute. So you count the number of the sucrose molecules here, right? Now let's look at this picture. Definition here. Now, solution A has a lower solute concentration. Okay, so let's see why lower. Now look at A. A on the left hand side, you will see there's only one. Okay, there's only one what you call that, one sucrose molecule, whereas on the B you get three. Okay, and then if you talk about solute, A will be less, okay, and B will be more. But if you talk about solvent, look at the blue circles here, or the blue molecules here. They are referring to the water. So for A, let's look at A only, blue ones. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I've got 10. I've got 10 molecules of water on the left, on the A. Whereas on this side, 1, 2, 3, 4, 5, 6. I've got 6 on the B. Okay, so if you're talking about solute, normally we refer to solute, right? So solute, solution A will have a lower solute concentration, but you see it's got more water. So you see that's why I say higher water potential. Higher water potential means it's got more water. That's all. That's what it means by water potential. That means the concentration of water is higher. It will have less solute, one red one. The other side got three. But in terms of water, it will have 10 here. Okay, the other side, the B will have only 6. Okay, so you can also put it the other way around. Solution B has a high solute concentration because there are 3 there, and has a low water potential because there are only 6 compared to the other side. Okay, so you see if it's high solute, that means you will have a low water potential lah. If it has a low solute concentration, it will have a higher water potential. Okay, it's the same thing. You cannot be high in water and also high in solute. You can only be high in water, that means you're low in solute. Okay, or you high in solute, you're low in water. All right, okay. So explanation. So now we say if you compare A to B, A is considered a hypotonic solution because A has a lower solute concentration. A only got one red one, okay, red, you know that solid here, and it got more water. It's got 10 molecules of water there, right? So A is hypotonic to B. Okay, therefore, because of this difference here, only water will be able to allow this to pass through the selectively permeable membrane due to the size, right? So you will find that there's going to be a net movement of water from A to B. Okay, the solute will try to go through, will try, but it cannot allow, it's not allowed to because of the size. So as a result, only water will be able to pass it. So as you can see that in the end, the resulting effect you will see is water will move from A to B. So water diffuses from solution A to B by osmosis. Okay, so the process of water movement is called osmosis. All right, okay. So always from hypotonic to hypertonic. The movement of water is always from hypo to hyper. Okay. Now, next one. So when talk about hypertonic solution is the opposite lah, right? Now you will see that a hypertonic is a solution that has a higher concentration to another solution, which is opposite concept only, where you must look for now more solute but less water. That is hypertonic. Okay, let's look at this one. In this case, you find that solution A will have more solute, which is got 3 there, right? B has only got 1. And then water will be less. Water only got 6 on the left, on the A side. And then B will have 10. All right, so solution A is said to be a hypertonic solution. Okay, so solution A has a high solute concentration and a low water potential. So if it's high solute, that means low water. Okay, or if the opposite here, the B will have high water but low solute. Okay, and explanation, the other way you can also use the other word. Solution B has a low solute concentration and a high water potential. Okay, so that means A is hypertonic to B. Okay, or you can say B is hypotonic to A. Okay, either way you're comparing both solutions side by side. A is hypertonic compared to B, whereas B is hypotonic compared to A. Okay, so as a result, you find that this movement of water, the movement will be from B to A, okay, through osmosis, because always from more water will move to less water. All right, so it's always from hypotonic. The movement from water is from hypotonic solution to hypertonic solution. Okay, so here B to A by osmosis. Okay, clear? I think there are no questions here because it's easy, right? All these things you probably, your school has done it, or maybe a teacher has taught it before. Okay, so now the effect of hyper, and isotonic solutions on animal cells and plant cells. This is an important concept, yeah? They always ask you, what happens if I put this animal cell or plant cell into these three types of solution? What is going to happen? Okay, so let's look at that. Now, the fluid found in the cell, that any fluid inside the cell is known as intracellular fluid. Okay, the fluid that is inside the cell is intra means inside, right? Okay, inside the cell. And the fluid that is found outside the cell is called extracellular fluid. Okay, it makes a lot of sense, right? So now, cytoplasm, the cytoplasm will contain, of course, it cannot be just pure water, right? It has to be, of course, it has got substances that the cell needs. For example, amino acid, glucose, minerals, salts, okay? Or metabolic processes, metabolic substances that the cell needs. So it has to have solute inside. Okay, a cytoplasm, whether it's a vacuum or not, it will always have substances inside there. So it's not pure water, right? Whatever that's inside the cytoplasm, it has substances, diluted substances, all right? So now the movement of water, whether water moves in or water moves out, depending on which side, okay, of the cell has got more water. Okay, whether it's outside the water, outside the cell, whether it is hypotonic or whether it's hypertonic, all right, that will affect whether the water goes in or the water goes out. And then if the water goes in, then your cell will expand, all right? The cell will become bigger and bigger and bigger, and it might even explode, actually. It might even rupture, okay, if it doesn't have a protection, okay? So we're going to see these cases now. What happens if we put different cells, animal cell and plant cell, into different solution, okay? And then you must know how to explain it. Okay, so let's look at, first of all, red blood cell. Okay, red blood cell, yeah. So we're not looking at other cells, but either the effect will always be the same, okay? Because animal cells do not have cell wall. If water happens to enter the cell in large amounts, find that the cell will bloat up, all right? And if the pressure is too great, or the cell will rupture or it will burst. Okay, let's look at hypotonic first. Let's see what happens if I subject a red blood cell in a hypotonic solution. Okay, I can put it in pure water, let's say distilled water. Distilled water is always hypotonic because there's only water there. Okay, there is no mineral dissolved inside the distilled water. So distilled water is always hypotonic solution. Okay, when red blood cells are placed in a hypotonic solution, okay, let's say, yeah. So outside, the water there is called hypotonic. Okay, it's high water potential. Hypotonic means there is very little solute. There's a lot of water, right? High water potential. And then comparatively, inside the red blood cell, because there is minerals, there are substances, there are solutes and all that, it will be considered hypertonic by comparison. Okay, so the red blood cell will have a higher solute concentration, or we call a lower water potential than the hypotonic solution. Okay, we say it's hypertonic because it's got more solute. And then again, I told you, like, when there's more solute, it cannot be more water. When more solute means less water, so low water potential. All right, outside more water, inside less water. So you can see the difference here. Outside more water, inside less water. So water always moves from hypo to hyper. Okay, remember that, osmosis. More water will move to less water. High water potential move to low water potential. That means water will rush into the cell. Okay, water will diffuse into the cell by osmosis. So water will move in, right? Water comes in. Just water, okay, water comes in, and the cell will swell, bengkak, right? It will be bloated up. It may even burst, rupture. It will burst when the pressure becomes too great. Okay, the cells will burst, it will swell up and then it bursts. All right, then when you draw bursting up, please make sure you draw the cell not intact. That means broken. It's important how you draw it, because we must know how to see. You must know how to show that the cell is broken. Sometimes just simply draw a simple shape doesn't look is broken. It looks like it's shriveled up. It may not show the swelling. Okay, so this is another diagram you can use, okay, to show that it's broken, or you can also show like this, or you can also show is incomplete like this. Okay, this is how you draw lah, right? Okay, the reason here is because the plasma membrane is too thin, okay, to withstand the pressure built in the cells. So the bursting all this phenomenon is known as hemolysis. Now remember, this word hemolysis is only used for red blood cells, okay, because of the word hemo referring to the hemoglobin. All right, you do not call this hemolysis for any other cells. Let's say you put amoeba inside there, right, in the hypotonic solution, water moves in, all right, and then let's say the contractile vacuum does not work, let's say lah, okay, but then your cell will burst and you call it hemolysis. No, you don't call it hemolysis because it's not red blood cell. Right, you only use it for red blood cell. Okay, so this is what happens. Now you must know how to in sequence. You must know how to explain what happens. First of all, you always mention the condition. Where is which side of the membrane? Is it hyper or hypo? Right? And as a result, water diffuses into the cell by osmosis. You must mention the word by osmosis, and then explain the condition. How does it look like? Okay, is it burst? Right? Has it burst? And the name of the phenomenon. Okay, what happens if you put it in hypertonic solution? Okay, you put in a solution that is too concentrated in terms of salt, right? Now, when the red blood cells are placed in hypertonic solution, so this is hypertonic solution. When it's hypertonic, means too much salt. Okay, high concentration of solute. Then that means water potential be lower. Okay, high solute but lower water potential. And then by comparison, if you compare outside it's hypertonic, that means inside the cell here will have it will be considered higher water potential. We call it hypotonic. Okay, there is salt there is salt, but it will be less compared to outside. Outside is higher salt content. Okay, so RBC has lower solute concentration or a higher water potential than the hypertonic solution. So by comparison, in the red blood cell, it looks like if there's more water compared to outside. Okay, so now remember what I told you about hypo to hyper, movement of water. So as a result, the water will diffuse out of the cell by osmosis. So water will move out. Okay, and then that causes your cell to shrink. Okay, so you can use this word shrink, shrivel, or crinkle. Okay, it's like like a prune. It looks like a dried, what they call that, the kismis, the dried grapes. I call it the currant, black currant, or the raisin. So everything all bunched up together, and it is all become smaller. And sometimes you may even draw like a durian like that. Yeah, okay, I call it a durian. Yeah, durian shape, all crinkle up like that. Okay, and it looks like that. All right, so you must know how to draw. Make sure that it's not rounded. You must not draw the biconcave disc shape anymore. You must draw it like all shrunken inside. Okay, and this process is known as crenation. Okay, crenation, the process of the thing bunching up and crinkling or shriveling or shrinking. Okay, now that is what happened when the cell is put in the what you call that, the hypertonic. All right, what happens if it's isotonic? All right, if your red blood cells are placed in isotonic solution, that means both sides, whatever solution that is placed inside, it has the same concentration with what is already inside your red blood cell. Okay, so the red blood cell has equal solute concentration or equal water potential as the extracellular environment. Same, both sides have the same concentration. So as a result, okay, water of course will go in and out. Yeah? We cannot say osmosis stops actually. Yeah? Osmosis will still happen, but the water will diffuse in and out of the cell at the same rate. Means how much water that goes in, how fast water goes in will be the same as how fast the water goes out. So there's no effect at all actually. The shape, how much water goes in and out is about the same. So it doesn't affect the shape of the cell. It doesn't change the shape of the cell at all. Okay, so the cells will maintain their normal shape. So you draw the perfect shape of your this biconcave, this shape, slightly indented inside the middle, in the middle part of it. Okay, the cells will maintain their normal shape, and there will be no net movement of water molecules across the plasma membrane. You cannot say no movement, not no movement, no net movement. It makes a difference. No net movement means there is still movement, but how much goes out, how much goes in is the same. Okay, you cannot say no movement. No movement is wrong. There is still movement. All right. Okay, now what about plant cells? Okay, what is the effect of hyper, and isotonic on plant cells? Okay, so now we look at hypotonic solution, the effect. Put into hypotonic first. The best example of hypertonic solution is always distilled water. Okay, put in distilled water. That is always hypotonic solution. Okay, so hypotonic solution will have high water potential. That means like you put in distilled water, alright, that's pure water, no minerals at all. And then the cell will have a higher solid concentration. Inside the cell, there will be solids. Inside the cell will be amino acids and glucose and sugar and whatever it is. That means inside here will be higher solid concentration. There will be less water, lower water potential than the outside. Okay, so now you have more water outside, less water inside. All right. So as a result, yeah, hypo to hyper, water will diffuse from the concentration from high water potential to low water potential. Okay, so water will diffuse into the vacuum by osmosis. So water moves in. So okay, so your vacuum will expand. Your vacuum will become bigger. Your vacuum becomes bigger and it will push the plasma membrane to the cell wall. So it pushes it, there will be slight pressure, right? And this kind of pressure is called turgor pressure. Turgor. Okay, and this is important for plants that do not have wood, right, for support. They need this water pressure to maintain the turgidity of the cell so the plant does not become wilted. Okay, they need this kind of pressure. That pressure is called turgor pressure. Okay, and now this cell is considered as turgid. Turgid means there's pressure inside. It is not soft, okay? It's not flaccid, right? So now the cell becomes turgid. Okay, there's pressure pressing onto the cell wall. The cell does not burst, even though there's pressure there, but it does not burst because there is a very strong structure there that makes up the cell wall. There's the cellulose. The cell wall is rigid, so it can withstand the pressure. The plasma membrane is stuck to the cell wall, but it does not make the cell burst because of the presence of cell wall. Okay, so this turgor pressure maintains the shape of the cell. So the cell remains this shape and gives support. This is for especially herbaceous plant. They rely on this support from the turgidity of the cell to make sure that the plant can stand up straight. Okay, it does not wilt. Okay, it does not droop, right? The plants. Okay, then it also cell turgidity also allows the gut cell to open. Actually, the gut cells will bend, and this causes the stomata to open. This one you learned in Form 3 before, okay, last year, but maybe have forgotten that. You read it, you learn it again, all right, next year. So this cell wall, right, I mean the turgidity causes the gut cell to swell, and that allows the stomata to open, and that is to allow gaseous exchange for photosynthesis and also respiration to happen. Okay, so that's hypotonic. Now let's say hypertonic. What happens if you put into hypertonic solution? So hypertonic, low water potential, you put in very salted water, right? Let's say, and as a result, yeah, you can see the compare first. Inside the cell will have a lower solid concentration because outside is higher. Lower solid concentration means there is more water inside, higher water potential. Okay, higher water potential in the vacuum. And that will cause now the water will diffuse out because inside higher water potential. Inside is hypo, outside hyper now. So water will rush out, okay? It will diffuse out the cell by osmosis. So always remember how you answer these kind of questions is you compare both sides first. You say which side hyper, which side hypo. Then the next sentence is you say the movement of water. Water diffuses out or water diffuses in by osmosis. Okay, and then next one you talk about the effect. So what happens to your cell? Imagine the water goes out. Okay, so the water goes out, your vacuole and your cytoplasm will shrink because the cytoplasm is so made of water. So lack of water will make the volume of cytoplasm will become smaller. So it's going to shrink. You can see this next picture here, at the bottom here, plasmolyzed. The yellow part here is actually referring to the volume of the uh, cytoplasm. See, it's so small, right? The cytoplasm has become so, what do you call that? See, this part here. And all this empty space is already filled with the hypertonic solution. The hypertonic solution has gone inside here, okay? It's gone inside here. This white part here is actually your hypertonic solution. And then your cytoplasm has become so small already, the size. Okay, so this causes the plasma membrane to pull away from the cell wall. Originally, the plasma membrane is stuck to the cell wall, okay, because of the pressure of the water. Now the water is gone, so nothing to push this plasma membrane to stick to the cell wall, so that's why it will collapse. So like the cell, the volume, the entire cytoplasm will become smaller, the membrane will pull away, so that's why it kecuk already, okay, shrunk. So now you can see here, this is the this line I'm drawing the red color. This one referring to the plasma membrane. Okay, you cannot see earlier because it was stuck to the cell wall. But now because it shrunk, now you can see the line already. Okay, so this phenomenon is known as plasmolysis. Okay, so the whole thing has shrunk and we call this this phenomenon is called plasmolysis. Alright, now plasmolysis is what causes the leaves and the stem of the plant to wilt because there's not enough pressure there. So that's why when the very hot day, right? When your plant is not properly watered, there's not enough water in the soil. Okay, after prolonged hours of transpiration happens, and then you find that the plant may even be drooping. The flowers will droop, or the leaves will be drooping because the water pressure or the hydrostatic pressure, the turgidity, the turgor pressure inside the plant has has dropped, has less, it become less, so it cannot support, right, the stem, so it all becomes wilted, right? Okay, now you can see in under the microscope, this is what happens when the cell is turgid lah. You see the pink color is labeled, this is cytoplasm, you just color it already, okay? Now if it is undergoing plasmolysis, you will see this, picture. You see that the cell wall is quite obvious. I'll put black color here. This is the cell wall, right? But the pink color, this is the volume of the cytoplasm, has already shrunk. And you can see it pull away from the side. It pull away from the side of the cell wall. Okay, so when you see this, what you're looking at is actually plasmolysis. Okay, plasmolysis they're looking at. Now, let me introduce you another concept now. Now you can reverse this, If your plant has undergone plasmolysis, you can reverse it in time. All right, if you can put it back into hypotonic solution in time, this process can be reversed. That means you're going to bring back water into the cell, but not too long. If the cell is under this condition wilted for too long, all right, it could be irreversible, cannot reverse anymore. That means the plant will die. But if it's put into hypotonic solution in time, you can reverse the condition. So now you got to put it in hypotonic solution. So the best is to always put in distilled water. All right. So comparison, if you compare, in outside is hypotonic, now inside compare is hypertonic because there's still salt inside there. So what happens is now water will diffuse into the vacuole by osmosis. Okay, you reverse it. You put into hypotonic, water now go back inside. Okay, and then the vacuole will expand. All right, it will expand and it will push the plasma membrane back to the cell wall. So you don't see this line anymore. You will not see this line anymore. Okay, you won't see this anymore. You will look like the cell will go back like normal. Okay, you see the line it will go back and press against the cell wall. The plasma membrane will be pressed against the cell wall. Now you reverse the condition. The cell will become turgid again. There's water inside already, the vacuum. It becomes turgid. There's turbo pressure. This process of reversing it back to, just now was plasmolysis, right? Now you're reversing the plasmolysis. You call it deplasmolysis. The process that you have done now by introducing water back into the plasma membrane, into the cytoplasm, this is called deplasmolysis. Okay, and if you look at the picture, this was just now like this. All right, when water goes back, it will become like this again. All right, water will move in, it will expand. All right, the vacuum. Okay, clear. Now, next one. What about isotonic? Okay, simple lah. This is just like simple as what we did just earlier. So when you put in the isotonic solution, both the solution outside and inside the cell are the same concentration. The water potential is equal inside and outside. Both sides have the same amount of water, right? So you consider as isotonics to each other. So the water will diffuse in and out by osmosis at the same rate. Right? Both sides, same rate, yeah? All right? Same rate moving. How much going in and how much going out will be the same, at the same rate. Okay. And then this one is the same picture, right? Showing you. So there's no net movement. Again, I stress the word net, yeah? You cannot say no movement. No net movement of water molecules across the plasma membrane. All right. And cells maintain their normal shape. So you draw the cell like this. All right. Okay. So, and the cell looks like this. Okay. All right. Okay. So this is the end of my lesson. All right. So I will not go through the 3.4 because this is already quite a lengthy class. So I will continue 3.4 next week, hopefully. Yeah. So I will try to finish 3.4 by next week, and the following week I will try to do chapter 4 then. Okay. So all the best to you, and thank you for watching my class. And if you find that your friends may need this lesson or revision or something or explanation on certain concepts, you can share my this the they call it the link. You can even share the channel. How to score in biology? Okay. You have to know your facts, okay, and you also need to know your technique menjawab. Okay, knowing the facts is one important hurdle you must cross that. Okay, in form 5, yeah. How to score? You need to know how to answer questions. Now that will be the person who best guide you will be your teacher in school, because she will be seeing you most of the time. She'll be giving you exercises, and what you need to know is ask the teacher what is the best way to answer this question. Okay, you can also ask me. I will message you through Telegram now, right? But you need to know by doing questions. All right, it is not by asking. You have to do the questions, and then when you get your answers marked, then you will know, oh, I think I know the concept, but how come I'm getting so little marks? Then the teacher will tell you, okay, because you did not write in this way. Okay, so that's why in order to score, you have no other choice, but you have to do questions. You have to do questions. By memorizing is only 1/4 of the battle. Not quarter, 1/3. I would say three parts of it. One is to memorize your facts. Number two, it is actually the first thing is to understand. Yeah, most important. 1/3 of it is understand. That's what I'm here for, right? The second part is to knowing, to remember, and that one I cannot do for you. All right, that one I cannot learn for you. I cannot read for you. Right? I can make you understand, but I can't remember for you. That one you have to do. And then the third part is answering questions. Answering question is you have to do exercises and you have to submit to the teacher. The teacher will mark for you. All right. If it's wrong, the teacher will tell you, okay, you did not write the correct way. That's why your marks are so low. Even though you think you write the right thing. The facts are there, but you didn't write in the proper way that the scheme wants. That one you have to come by experience. Okay, that one you cannot just ask. You have to do. Okay, all right. Okay, so I hope that answers your questions. So I hope to see you next week then. All right, please share my link and share my channel to people who might need it. Your friends probably, all right, or maybe your brothers, sisters, or anyone who might be doing biology next time. All right. So here is my Telegram name if you would like to get in touch with me. All right. So please do. I will try to answer your questions if I can. All right. Okay. Bye bye, everyone. See you. Okay. Thank you. Bye bye. See you next week.